Hi, and welcome to our lecture on the senses. In this chapter, we're going to ask the question, does this person have severe allergic conjunctivitis, or are they just really high? So let's go ahead and begin with the idea that if we're going to have a set of senses, we need some kind of reception of information about the outside and occasionally the inside world. And we're going to begin here with what we call chemoreceptors as one of our types of receptors. These detect specific molecules. An example of this could, of course, be detecting something like carbon dioxide levels within your blood, maybe detecting pH or oxygen levels. And we can even think about chemoreceptors as the kinds of things that respond to molecules floating through the air, which eventually we might interpret as smell. Now we do, of course, have a lot of different kind of receptors, so let's take a look at a few others. Nociceptors detect chemicals from damaged cells. We interpret these signals as pain, so nociceptors we sometimes refer to as pain receptors. And they're kind of different than chemoreceptors because they are generally recognized as naked dendrites, but they are going to pick up these chemicals released from our damaged cells. We also have osmoreceptors, which are going to detect changes in solute concentration within our blood plasma, and this obviously is going to be very important. We get the idea that we might have to stimulate thirst, let's say if things are hyperosmolar, if we have too much dissolved substance and we need more water. We can also relate this to changes in blood pressure, so we can constrict or relax blood vessels as we see blood pressure change, and of course that can be associated with differences in solute concentration. We also have thermoreceptors, which detect changes in temperature, and of course we're going to find these in places like our skin. And then in our retina we have photoreceptors, which detect light. Now we can break these up, as we can see in this particular picture, into cones and rods, and the kind of simple difference between these is that we have multiple kinds of cones, and they respond to a narrow range of wavelengths of light. Therefore, we can use them to determine color. So we have this 3-cone system in humans, which is going to allow us to pick up a relatively broad range of colors, although in general, with exceptions of some people with specific receptors that are kind of, let's say, not common, those kinds of receptors might actually pick up some of the UV spectrum as well. I think that they've only been found in women, but we also see those kinds of things in birds. Rods, on the other hand, are really going to be more about picking up light or not picking up light. And we're going to use these for some kind of night vision, but they're going to lack the ability to differentiate between colors. Mechanoreceptors are a little bit more difficult to talk about in some ways because they come in a number of varieties. So we'll recognize mechanoreceptors in general as those that detect physical changes, and this does result from compression or stretch, so that there is something in the environment that's causing them to elicit that stimulus. It helps make sense of them if we take a look at a few different classes of them. So if we begin with tactile receptors, these are the ones found within our skin, which are going to detect touch or pressure. So when we grab a hold of something, let's say with our hands, we know that we're doing that because we can feel that object and we can feel how much pressure we have on that, because there are different kinds of receptors. We then have baroreceptors, and we see some examples of locations of those in the image here. And those are going to give us ideas about kind of the stretching associated with various things like our lungs and our intestines, to let us know kind of what's going on as far as the volume of these things. Now, some of the interesting examples of this would of course be with urination or defecation, where when we detect that stretch, there's going to be some communication with the spinal cord, causing what we refer to as a urinary or a defecatory reflex. Then we have the limb positioning proprioceptors. So proprioceptors are really cool in that they let us know where our limbs are in space because of the detection of stretch of our tendons. And so if you think about this, you can place your arm behind your back, you could be blindfolded so you can't see it, but you know where your limb is in space. You can close your eyes and probably touch your nose using your hand and your arm because you know where your limb is. So these proprioceptors are really important for our capacity to use our body, but particularly our limbs. Now we're going to go ahead and jump into a couple of different kinds of pain which do help explain a little bit about how our sensory system works. We're going to take a look at phantom pain here, and then we're going to take a look at what we refer to as referred pain. So phantom pain is the perception of pain in an amputated or excised part of the body. And so we can see the example here from Monty Python and the Holy Grail where the Black Knight maybe is missing some limbs, yet it might be possible to still feel pain or itching within that limb which isn't actually there. And so we kind of want to think about why this would be. And this happens because the nerve bodies, the cell bodies of those neurons that were serving that limb and receiving that sensory information and bringing it back to the spinal cord, well, those cell bodies still exist. And they remain alive and can propagate impulses that go to the central nervous system. Now, of course, when someone amputates a limb, they try to make sure that they treat the nerves in such a way that they're not going to have these involuntary stimulations. But the reality is, with limb loss, there does tend to be some form of phantom pain. And of course, it's problematic because the limb isn't there. You don't have a problem. You're not releasing chemicals from damaged cells. So the pain is really inappropriate. Referred pain is interesting in that it is diagnostically useful. So referred pain is where we get pain from organs, that is visceral pain, but we experience the pain cutaneously. And of course, we did contrast in the Introduction to Nervous System chapter this idea of visceral sensation versus the somatic or cutaneous kind of reception of information. So in this case, we're going to be thinking about some particular patterns of this and how this can be useful. The pain from the liver and gallbladder often manifests itself in the right shoulder. That seems completely inappropriate, right? Because the liver's not in that area, so you're not confused between cutaneous pain or visceral pain within the same location, but rather it's occurring in an entirely different kind of location. We also see a pattern that appears in males more so than in females, where we have pain associated with the chest that then radiates down the left arm associated with heart pain. So again, this doesn't make sense to have pain in the left arm as a result of a heart attack, and this can be a signal of a heart attack. One of the interesting things about this particular pattern of referred pain is that people used to dismiss heart attacks in women because they weren't exhibiting this symptom. But it ends up that studies show that this particular symptom isn't actually all that common in women, even though it does tend to be rather common in men. But it's just a reminder that when we do studies, it's really important to look at lots of different groups because we may see different responses. Whether those different responses come from different ethnic groups, whether those different responses come from males versus females or relative to age, we have to make sure that after initial studies, we expand our studies to make sure that whatever pattern we're seeing actually holds up for all different groups. Otherwise, we may not provide proper medical care. Question we might have about this is, why would we see these kinds of patterns? Why, for example, might we get appendix pain relatively close to the center of the body rather than over where the appendix is located? And the thought is that neurons from the same spinal segment are going to be basically associated with each of the damaged organ and the cutaneous area where stuff is felt. If we remember back to when we learned about the spinal cord, we know that sensory neurons are going to travel on the spinal nerve, then on the posterior root with their cell bodies in that posterior root ganglion, and they're going to enter the posterior horn of that spinal cord. So what we're talking about is having the sensory neurons from a cutaneous area, like the area around the umbilicus, go into the same area that the visceral sensory information is going from the appendix, and therefore they may be synapsing with the same interneurons, and we can't discriminate between pain in those two different areas. So here we can kind of get a really good look at what we're talking about. And again, we have our cutaneous information coming from the skin. We have our visceral information. The visceral information has to travel through the sympathetic trunk ganglion on that white ramus to get on the spinal nerve. We're going to come directly back on that spinal nerve from the nociceptors here, but they are both heading to the same general area of the posterior horn of that spinal cord. Therefore, we maybe can't interpret the difference between those two areas. All right, we're going to go ahead and now jump into special senses. Special senses, of course, are some of the somatic senses, things that we are conscious of. And these special senses can be kind of broken down into taste, smell, vision, and hearing. We're not going to spend a lot of time on taste and smell. We're going to be focusing mostly on vision and hearing. We do refer to taste as gustation. That's going to be the technical term we use for that. And we do associate this with two cranial nerves, facial nerve and the glossopharyngeal nerve. So that's nerves numbers 7 and 9. Now, so that means that we're going to have these taste buds located along these papillae, these bumps that are on our tongue, and that they are going to respond obviously to particular chemicals in here, and that's going to lead to a nerve. This would be a branch of one of those cranial nerves. So we get an idea of how this is going to work as we have our two nerves coming from our tongue. So we can see the facial nerve coming up from the front part of the tongue. From the posterior part of the tongue, we have that glossopharyngeal nerve. They come into the brainstem. They send information to the thalamus. The thalamus will then direct that to the gustatory cortex, which we associate with the insula. Olfaction is, of course, our technical term for smell, and we do only have a single nerve for this one. In fact, it's nerve number 1, the olfactory nerve. And if we think about how this works, we have our olfactory nerves down in here, ending in what we refer to as olfactory hairs, which can bind to particular odor molecules, and those nerves are going to fire when they do so. Of course, they run up through the cribriform foramina and synapse in an area we refer to as the olfactory bulb. This is a bulge that sits on top of the cribriform plate, and then the olfactory tract will carry that information back to our primary olfactory cortex. It is important to kind of recognize as we think about this that any particular smell is going to be whatever combination of firing olfactory nerves we have. Therefore, a smell could be relatively simple and be associated with a particular kind of olfactory nerve receptor, or it could be rather complicated and be associated with many of them. And that's going to take us into vision. We're going to go ahead and begin here with this really nasty case of allergic conjunctivitis. So the conjunctiva is a mucous membrane that's forming a lining over the eye, but not the cornea. So where we see the colored iris here, it's covered by a clear portion called the cornea, and the conjunctiva we can see nicely because it's so puffed up here, doesn't cover that, but it's covering the entire white of the eye. It's filled with blood vessels. It's going to help lubricate the eye, and it even lines the inside of the eyelids. So it is a kind of an important membrane here, providing some of the oxygen supply. This one, again, super swollen. Conjunctivitis, or the inflammation of the conjunctiva, can come from allergies, it can come from viruses, it can come from bacteria. So something that we might refer to as pink eye doesn't necessarily have one specific cause. We want to figure out what that cause is to get an idea of how likely it is to spread from someone to someone else. Obviously, we would worry about the spread of bacterial conjunctivitis and not of allergic conjunctivitis. So let's talk a little bit about tear production here. We of course know that the lacrimal gland, innervated by the facial nerve, is going to produce tears. It's located superolateral to the eyeball, and the tears are going to wash across the eyeball, obviously potentially washing things out of the eye, before draining out through what we call lacrimal puncta, which are on the medial side of the eye. So we get this kind of flow from lateral to medial, from lacrimal gland to the drainage system. The lacrimal puncta are this drainage system. If you take a look at your eyes close in the mirror, you should be able to see these little holes on that medial side in both your upper and lower eyelids. This particular diagram shows what we refer to as puncta plugs, which can actually just be a kind of simple manual mechanism for preventing the drainage of tears from the eye, and this is one possible treatment for dry eye. This was particularly used prior to the development of drops, which would stimulate more tear production. The tears will then drain through the nasolacrimal canal. If you remember your lacrimal bone, it kind of had a little groove in it with a hole in the bottom. And this nasolacrimal sac here is going to sit in that groove, and the nasolacrimal canal will drain down through that hole, causing tears to drain out into your nose. Now, I should, of course, while I'm thinking of the eyes, think of eyebrows, or in this case, eyebrow. And it's a little bit confusing to think about the function of eyebrows because they often don't do exactly what they need to do, but they should be blocking sweat from dripping down into the eyes. And you can see how a nice unibrow like this might work pretty well for that. Of course, if you are really, really sweaty, you're going to have to do something to kind of mop the sweat up out of these, but they can assist in it for a while at least of keeping sweat from dripping in your eye. Now, as far as the eye itself goes, this is a 3-layered structure. It's actually a really common setup in the body. We could treat our blood vessels as also having 3 tunics or layers. So we're going to have kind of a superficial outer fibrous tunic. And our fibrous tunic we see here is shown kind of in white. This front part would actually be clear here. It has only 2 parts, the sclera and the cornea. We have a middle vascular layer here shown in pink. This is going to have things like the iris, the colored part of the eye, which of course is muscular and can control the size of the pupil. There's going to be a ciliary body, which helps adjust the tension on the suspensory ligaments, which are attached to this lens here, so we can change focus. And they also produce the fluid that maintains itself in the front part of the eye. And back here, this vascular structure is the choroid. Then we have the deep neural tunic, which is the retina, located only in the posterior part of the eye, called the neural tunic because, of course, we're going to have our neurons coming from our photoreceptors, ultimately forming the optic nerve as that exits the eyeball. We'll begin here with that fibrous tunic and the cornea. The cornea is avascular. It is transparent, so light can go through this. It is not the lens of the eye, and that's important to recognize. It's on the outside here. You actually have to go through that hole there, the pupil, to find where the lens is. It does have its own epithelial layer. It shouldn't have capillaries over it because it is transparent and we want to be able to see through this. But we can have some issues with this cornea. Of course, it can be injured. I've had something stuck in mine that I've had to go to the doctor to get out. There is a heck of a lot of pain that can be associated with this. But you can see in this case that there are blood vessels that are actually growing into this transparent area, and they shouldn't be doing that. This is referred to as corneal neovascularization. That is obviously the formation of new blood vessels on the cornea. And this is one of the things that can happen if you wear your contacts for too long. You're probably aware that contact lenses have a degree of permeability to allow exchange of gases through the lens. And that's because the epithelial layer of the cornea is directly going to receive oxygen from the external environment rather than from blood vessels. Now, if you wear your contacts for too long, the porosity is going to decrease over time, and they're no longer going to be able to exchange gases. That means you are going to be depriving this area of oxygen, and blood vessels will start to grow through an area where they shouldn't have them. Of course, it's also possible to get ulcers and other things if you leave your lenses in for too long. So it's really important to kind of understand how long your particular contact lenses can remain in and to follow those recommendations. They're not just there to make you buy more lenses. The sclera, of course, is the white of the eye. One of the most important things about the sclera for us is that it is the insertion point for the extraocular muscles, the muscles that are going to move the eyeball around so we can look in various different directions. It is composed of dense fibrous connective tissue. It's going to be continuous with the cornea there. They again together form this fibrous tunic, and it has some openings here, what we call scleral venous sinuses, so that the fluid produced back here can flow out and onto the eye here. So we're going to go ahead and jump into the vascular tunic at this point. We're going to begin posteriorly with the choroid. You might remember that term from the choroid plexuses, which were also vascular structures. In that case, those were actually helping to form the CSF. But this is just the vascular area between the retina and the sclera in the posterior part of the eye. So it's going to lay adjacent to the pigmented layer of the retina, which will kind of help absorb light, and then this choroid here can provide nutrients to that deep layer of the retina. As we move forward, we get to the ciliary body. The ciliary body is a little bit complex. We say it's thickened because it's like the choroid gets kind of fat here as we get near this lens. And there's kind of a good reason for this, because this has a couple of jobs. It has a muscular ring which can adjust the tension on these suspensory ligaments, adjusting the focus of our lens. And it also secretes aqueous humor. Humor is just kind of a general term we use for fluid. The posterior part of the eye, everywhere kind of behind here, is filled with what we call vitreous humor, which is relatively thick or viscous. And the aqueous humor is the watery fluid produced by the ciliary body, draining through the pupil into the front of the eye and out through the scleral venous sinuses. So also associated with this vascular tunic would be the iris. The iris, of course, is this muscular layer here consisting of two basic layers. We're going to have the layers that are innervated by the sympathetic division of the autonomic nervous system, which will dilate the pupil to open it up bigger. And then the layer of muscle, which is associated with the parasympathetic division of the autonomic nervous system, which will constrict the pupil to make it smaller and allow less light in. Now, the pupil itself is actually nothing. It is literally the hole in the iris, and therefore the pupil isn't a thing. You can't put a tag on a pupil because it's the absence of iris, which means that when light comes in, it's going to travel through the cornea. It will go through the pupil. Anything that hits the iris should not make it through. Then it goes through the lens, and then that will direct it onto the retina, particularly to an area we'll get to in a moment. So the retina does form what we refer to as our neural tunic. It is the inside layer of the posterior part of the eye, and it's the part that contains the photoreceptors. And we can see a kind of a look at this under the microscope to get an idea of how organized this is. We of course have our choroid towards the outside, and outside of that would be the white of the eye or the sclera. You can see a pigmented part of the retina here. So we have all this pigment. And the importance of that is that our rods and cones sit in here. These are our photoreceptors. So you can imagine from the bottom here, light comes through. Weirdly enough, it actually goes through some ganglia. It goes through some nuclei of some cells, and it's going to hit the rods and cones down here. If it misses rods and cones, it's going to travel in through here and get absorbed by the pigmented part of the retina. You can understand why that's significant. If that wasn't there, some of that light would get absorbed in the choroid, but much of it would hit the white of the sclera and bounce back. And that would cause us to basically be able to see something on the bounce back, which would of course obscure the image. So the interesting thing is that the part that we're actually triggering is right here next to the pigmented layer. That's where we have our cones and our rods, and you'll learn more in physiology class about what happens when the light hits these and how that results in a chemical reaction, and how one of the reasons why the eye has to be well vascularized is that it essentially has to kind of reset these photoreceptors. All right, so let's take a look at the back of the retina and see what it actually looks like. We're really looking for a couple of different things here: the optic disc or blind spot, and the macula lutea, which has in the middle of it an area referred to as the fovea centralis, or the central focus. Beginning with the optic disc, you'll notice it is very different than everything else here. Most of this is kind of orangish, gets a little bit darker in this area of the macula lutea, but we seem to be having this reflection coming off of this optic disc. That's why this is the blind spot. Light that hits this will bounce off of it. This is going to be where all of the neurons are converging from all over the retina. So we've got neurons coming from here, mostly from rods. From the area in the macula lutea, we have information coming mostly from cones. But those neurons are coming here, and this is where all the neurons come together to form the optic nerve, which will then exit from the eye. And of course, those are myelinated neurons. Therefore, we're going to have this structure which is going to be reflective any light that hits that we can't see. That's why we refer to the optic disc also as the blind spot. It's just an area on the back of our retina where we don't get an image because we don't have any pigments or photoreceptors there to absorb the light and cause nerve transmission. Now, the neat thing about this is we can do some tests for our blind spot, but we normally don't notice it because we have two eyes, and of course our brain does a good job of kind of combining all that information together and filling in any gaps that we have. So we actually have to specifically test for the location of that blind spot, but we can find it. The macula lutea then is a highly pigmented area. It's essentially the exact opposite of the blind spot or optic disc. The optic disc is where we have no photoreceptors. This is our highest density of photoreceptors, and particularly of cones. The other thing that we know about this area, and the fovea centralis, the middle of it, the area that light hits when we're actually looking at an object, is that we have relatively few photoreceptors per neuron. So we can kind of think about that as being analogous to our motor units. We knew that if we had few muscle fibers per neuron, we were going to have better control. In this case, we have a higher definition image in the macula lutea and fovea centralis because we have fewer photoreceptors per neuron. So it's a high definition color image, and it is the darkest spot on the retina because it has the highest density of photoreceptors, which of course are pigmented and therefore absorb light rather than reflecting it as the blind spot or optic disc does. Now, one thing we want to think about when we think about the macula lutea is macular degeneration. And this would be where we have a form of blindness starting, in this case, at the macula lutea, which of course is the area that light hits when we're looking at an object. So you can see that for a person with macular degeneration, they actually have a hole in the central part of their vision. And then what they're seeing here, because they're seeing stuff with areas outside of the macula lutea or kind of on the edges of it as they start to lose it from the middle, is that their image isn't going to be as clear either. Kind of looks cloudy. It's not as high definition of an image because if you have macular degeneration, you are losing the best part of your vision. All right, so what about this lens? We want to think a little bit about the lens. We can kind of associate this with the vascular tunic because it is connected to the ciliary bodies, but really it's kind of a nice, unique component. It's strong, it's deformable, it's elastic, so you can kind of change its shape and it'll go back into a resting state. It is transparent and it's biconvex. That means it bulges out on both sides. You can see that it's kind of bulging out there and it's bulging out here. It's not really a sphere, but it's like a sphere that has been stretched by these ligaments here so that it's a little bit elongated. The neat thing about this is that it does have this connection to these suspensory ligaments. And the very first thing we want to kind of understand here is that these are called suspensory ligaments. They are not tendons. And therefore, when we think about the relationship they have to these muscles here within the ciliary body, what we call the ciliary muscles, that relationship is going to be a little bit complex. If we were to think about them as being tendons, we would imagine that as those muscles contracted, we're going to pull on those tendons, and that would pull on the lens. And that's not what happens here, because again, these aren't tendons, these are ligaments. And what is really going on is we have a difference between our distance vision and our close-up or near vision. So if we want to think about kind of what's going on here and to describe this, we need to take a look at the differences here. First thing you'll notice is that we are quite bulged out here when we are focusing on objects that are close to us, and we have these kind of relaxed looking suspensory ligaments. We have here some tight suspensory ligaments when we're viewing an object at a distance, and the lens is relatively flattened in comparison to when we're looking at objects that are up close. So if we're looking at objects up close, the weirdest thing about this is that our muscles are contracted. But you can actually test this by looking at objects where you have to see them up very close, very small objects, trying to thread a needle, reading really small text. You're going to feel that your eyes can get tired, so they can get fatigued because you're actually contracting the muscles during this time. If you're viewing things farther away, the muscles are relaxed. So this is actually going to allow for tension on those ligaments. This pulls the lens into a flat position. Now there is actually something really interesting about this besides this kind of weirdness of contracting muscles to relieve the tension on the ligaments. And that is what happens when you get old. And I can speak of getting old as an old person. So generally somewhere around the ballpark of 40 years old or so, people start taking an interest in getting some reading glasses. And there's a reason for that, and it may not be exactly what you would think. So in order to see things up close, we need to contract our ciliary muscles. Now, I can contract my ciliary muscles. That's going to work just fine. When we do contract our ciliary muscles, we relieve tension on the suspensory ligaments. I can do that just fine. When we relieve tension on the suspensory ligaments, this relatively flattened lens, which is kind of flattened in our normal kind of throughout the day looking at distant objects kind of period, is going to bulge into this fatter shape here with a larger diameter. That's what I can't do. I can contract the muscles, I can relieve the tension, and my lens just stays in the normal flattened position. Again, it's not the normal state for the lens. The lens wants to bulge out, but when it's under tension, it stays kind of flattened. It just ends up that when you get older, your lens stays in that flattened position, and you relieve the tension, but because it has lost its elasticity, the lens doesn't kind of bulge out into that fatter shape for viewing objects up close. All right, so in addition to taking a look at the lens, we want to think about a couple of spaces in the eye, the posterior cavity and the anterior cavity. The posterior cavity is going to be posterior to that lens. It contains a gelatinous, stagnant vitreous humor, which we sometimes refer to the vitreous body because it's sort of like you've got kind of a blob of jelly back there. This is formed embryologically and it maintains itself. It's not something that we're constantly rebuilding. Anteriorly, we do form the aqueous humor in those ciliary bodies. It does flow through the pupil and out through those scleral venous sinuses. So this is aqueous, meaning it's watery, and it is recycled. We make it and then we let it drain out of there, so it has that kind of similar kind of under constant replacement status that we had for our cerebrospinal fluid. All right, and here we want to take a look at the optic chiasm, thinking about what happens when we take visual information back to our brain. We already know that we want to take information from our retina and deliver that back to our occipital lobes where we're going to process that information. And it's not quite as simple as you would think. You can see here that we've got this brain and we're bringing information back. This is the left eye. I know it's on the right side of the screen. That kind of indicates that we're taking a look at this brain from below. We've got this kind of left portion of this retina coming back and working its way to the left occipital lobe. But the right portion of the retina of the left eye is going to cross at the optic chiasm or optic chiasma, both of those terms are used, and that's going to head over to the right side, joining the right side of the right eye. So we're really crossing over a portion, about half of the visual information, to the other side. Now, of course, there are some other complexities that are happening here. You can see that we make some stopping points at the thalamus, and even prior to that, we have these stopping points at the corpora quadrigemina, so that we can kind of create that 3D kind of space around us that integrates vision and sound. Speaking of sound, that takes us to the ear, and relative to the eye, the ear is somewhat simple, although we still do break it up into 3 different regions, the external, middle, and internal. The external is going to be everything going up to the tympanum or the eardrum. So that's the external auditory canal and the auricle or pinna, this outer flap here. The middle ear is the area that houses the auditory ossicles, the malleus, incus, and stapes, or the hammer, anvil, and stirrup. It's also going to contain the Eustachian tube or auditory tube, which connects to the nasopharynx to allow for exchange of pressure. And then we have our inner ear, which is this fluid-containing area here with the cochlea for hearing and the vestibule and semicircular canals for equilibrium. Those, of course, are going to give rise to the vestibular and cochlear branches of the vestibulocochlear nerve, cranial nerve number 8. So let's take a look at these various parts, beginning here with the auricle or pinna. This is this flap on the outside. Of course, some humans have some muscles that can help them wiggle their ears, which are kind of the remnants of the muscles that are very common in mammals that help them kind of direct these ears in different directions to help them localize sounds. It ends up that for the most part, primates have lost some of that capacity, particularly humans, although we do sometimes retain some vestigial muscles there, again, allowing us to wiggle our ears, but not really to do a good job of localizing sounds. When we go through that external auditory canal, we end up at the tympanum. It is going to be the boundary between the external ear and the internal ear. Just want to point out we have this obviously in other organisms. This frog has that right on the outside of its head. They're, of course, going to have a simpler ear than mammals. We have a couple of additional bones in there that are derived from jaw bones evolutionarily. But this is a funnel-shaped sheet of epithelium. It picks up the sound waves and transfers those to the auditory ossicles or ear bones. We can see some issues with this. There's a nice malleus fused to this in a good, healthy eardrum here. This one looks less than healthy, right? We can see obviously some bleeding in here. Perhaps there's a little bit of a rupture that we can't quite make out because of that. And so obviously this person is having probably some very serious pain associated with this ear infection. Chronic ear infections sometimes are treated by ventilation or tympanostomy tubes, which can allow an exchange of pressure there so you don't have that intense pain associated with them. They will eventually fall out and that tympanum will heal over. Again, this should be in relatively good condition. We can see that malleus through the tympanum at that point. So the auditory tube is interesting. It's actually a horrible name. It's formerly known as the Eustachian Tube, named after a gentleman named Eustachio. This connects the middle ear cavity with the nasopharynx. It does allow for equalization of pressure. We can facilitate this through chewing gum or yawning, and so you're familiar with doing those kinds of activities as you travel, say, up and down mountains in your car. Obviously, if you're traveling up a mountain, you're going to get to higher areas where there's a lower air pressure. That means you're going to have this higher air pressure in your middle ear, and that's going to be putting kind of pressure from the inside on that tympanum. Again, if you can yawn, if you can open up that tube, you can potentially equalize that pressure. The opposite happens when we go down a mountain. So we go down a mountain, we get more air pressure, therefore the pressure is coming from the outside, pushing in, but the solution is still the same. So here are our auditory ossicles. We're going to label them from lateral to medial. Malleus, incus, and stapes. Hammer, anvil, and stirrup. This hammer obviously looks a little bit like a turkey thigh. The anvil does have this pointy area, and we have a horn on an anvil where we can kind of bend some metal. It's actually acting kind of like an anvil. This is acting like a hammer. The malleus is connected to the tympanic membrane, you can see here. Sound vibrations are placed into the malleus. It's going to push on the incus here. You'll notice both the malleus and the incus have these long lever arms, and those are going to magnify the sound waves, putting pressure on the stapes here, and that will kind of hammer in on that fluid environment of the inner ear to allow those sound vibrations to work their way down the cochlea. So we want to think about what structures then some of these bones are attached to. Obviously the incus is attached to the malleus on one side and the stapes on the other because it is in the middle. The malleus is the one that is most lateral. It's attached to the tympanum or the eardrum, and the stapes is attached to an area referred to as the oval window. We can't really see that here, but you might be able to make out that the base plate of the stapes is oval in shape. The oval window is a part of this vestibule here, as is this round window. And we do really want to think about why there are two windows, and we'll come back and see this, but this is a fluid environment here. Unlike the air-filled environment here, you can't compress a fluid. So to transfer the sound waves from this air-filled environment into this fluid-filled environment, you need to be able to push in on that fluid and there has to be a place here for this round window to bulge outward so that that fluid can actually move as we're transferring the sound waves in. All right, we do recognize what we refer to as the bony labyrinth and membranous labyrinth. We just kind of can think of this as where the ear is located in the case of the bony labyrinth. And the membranous labyrinth is then going to be a series of membrane which contain two fluids, this endolymph and this perilymph. We'll kind of come back to this idea that the inner ear is basically a set of a fluid-filled tube inside of a fluid-filled tube. But if you were to cut into the bony part of the temporal bone where this ear is, you would see kind of these squiggly things like little wormholes inside of the bone. That's the bony labyrinth. The membranous labyrinth then is the membranes where the sound waves are going to travel, or where we're going to get fluid traveling and putting pressure on these sensory receptors located in the ampullae, these expansions of it that help us figure out what's going on in regards to our equilibrium. So we're going to kind of break down the parts of this and recognize the different functions of these. We're starting here with the cochlea, obviously part of our vestibulocochlear nerve, right? We've used this term before. Cochlea refers to this snail shell shaped item here. You can see the spiraling of that. The spiral of that just allows a lengthy tube to fit into a smaller area. But we can imagine that cochlea unrolled and kind of being a nice long tube. That's important because different sound waves are going to travel different lengths down that tube. I do want to point out that students have a real problem with the word cochlea and spelling it. It is not Coachella. In fact, much of the stuff you hear there is stuff that really you probably didn't want to hear, or at least I didn't want to hear, and maybe doing damage to your cochlea, not because it's too loud, but just because it's kind of cruddy music. It's also not the cochlea. It's a co sound in the beginning, so the CO, and the CH sound is in the middle of it. But I just want to point out people have some struggles with this. And if you have been to Coachella, I just want to point out, yes, there have been a few good bands there. It does happen on occasion. All right, let's take a little bit of a look at how hearing works then. We want to kind of think about the concept of hearing from sounds coming in to us sending them out to our brain for processing. We have sound waves coming in through the external auditory canal. They have maybe been picked up by the auricle. We are going to vibrate the tympanic membrane, which will pass those vibrations through the malleus and incus, magnifying those before pushing on the stapes, which transfers those in through the oval window. We then travel down the cochlea here with high-pitched sounds only going partway down and low-pitched sounds going closer to the tip. Obviously, the more length of this you have, the more kind of differentiation you can do between sounds of different wavelengths. We're going to then cross these membranes in here, and we're not going to address the part in here, but this basilar membrane absolutely must vibrate. It's going to lead to the stretch of what we call stereocilia, which are attached to hair cells, and when they detect that stretch, that's going to cause our nerve transmission. That's kind of the simplified version of what's going on here. Now, of course, we said because we have to put these sound waves into this fluid environment, we need a place for them to go out, and we're going to have them travel here and bulge out at that round window. That's what allows us to transfer sound waves into a fluid environment, and hearing itself is really about the vibration of the basilar membrane as we cross here and work our way out of this. We, of course, also associate our ear and, of course, cranial nerve number 8, the vestibulocochlear nerve, with equilibrium. And there are two areas that are responsible for equilibrium. The semicircular canals, which conveniently are these three somewhat round tubes. So semicircular canal is a pretty great name for these. And these are responsible for what we call rotational equilibrium. When we think about having our head tilted to the side, and we know that our head is tilted to the side, that's that rotational equilibrium. We also have the vestibule, who gives part of our vestibulocochlear nerve's name, as this area that is essentially right here. It's the kind of input, so the stapes is going to push on the vestibule, even though those sound waves will work their way down to the cochlea, but it's kind of that middle area also between the semicircular canals and the cochlea. we can see that within that we have two areas, the utricle and saccule. We're not going to worry about identifying those, but those are the two areas that are responsible for what we call gravitational equilibrium. And gravitational equilibrium is going to be what we're often going to think of as acceleration or deceleration, and this can work in a couple of different ways. We have part of this functionings for like when your car accelerates and you feel that going forward, or decelerates and you feel that stopping. And then part of this is working when you feel the same kind of things in an elevator going up or down. So we've got the tilting, the kind of three-dimensional positioning of your body in space from the semicircular canals, and this idea of whether we're going up, down, forward, or back from the vestibule.